let me introduce Dr. Florian Krammer. He's a professor of vaccinology at the Icahn School of Medicine at Mount Sinai, and he will be chairing the next session. So I turn it over to you to introduce the first speaker.
So welcome to this session on basic research, and our first speaker for this session is going to be Dr. Lara Mahal, who is the Canada Excellence Research Chair in glycomics at the University of Alberta, and she's going to talk about glycosylation in influenza response and immunity. The floor is yours.
Okay, so I wanted to talk to you about our work in glycosylation and influenza response and immunity. And in the first half of the talk, I'm going to talk about our work in influenza response and how it corresponds to severity. And in the second half, I'll talk about some of our latest work in the vaccine area from the civics, where we're looking at glycosylation and how it may lay on to serum biomarkers for response to influenza. So next slide, please. So carbohydrates, so glycans, aka carbohydrates, or those sugars that people most likely chop off of all the stuff because they don't want to deal with them on the cell surface, are really, really critical to host response. And I think no audience knows that better than the influenza audience, because neuraminidase is a carbohydrate removing enzyme, and of course hemagglutinin is a carbohydrate binding protein, right? So if we think about it, carbohydrates are used to trap pathogens. We see this in the mucus, right, where you have sialic acids, you have these highly sialated mucosal layers that help to trap influenza. They're critical in enhancing the effector function and antibody response. We're getting more and more data that shows that the glycosylation of the FC region is really critical in determining how strong an immune response you're going to get. There's, of course, carbohydrates that are involved in the lectin-mediated immune response, so innate immunity, the lectin-mediated complement pathway, for example. And of course, carbohydrates can actually change the actual signaling of molecules and the strength of that signaling. And so these are just a few examples of the ways that sugars, so these complex carbohydrates, are really, really critical mediators of the immune system in ways that we may not always appreciate. Next slide, please. So my lab, next slide. I guess they're having trouble with the next slide. My lab really focuses on systems-based approaches to understanding the glycome. And to do this, we created a technology, which I'll talk about, lectin microarrays, and I'm going to talk about some of our work integrating data with it. So next slide, please. So what are lectin microarrays? It's a really simple concept. These are protein microarrays where the proteins are carbohydrate binding molecules. So we call them lectin microarrays because it's kind of a historical name, but nowadays they actually have antibodies on them as well. What we do is we take a fluorophore labeled sample, which is usually labeled on the protein backbone, so the carbohydrates are intact. We can add it to the array, we scan it, we get a glycopattern, and we can then analyze and back out from our knowledge of the carbohydrate binding proteins very specific carbohydrate signatures and molecular level detail. Next slide, please. So the information that is read by lectins is really encoded in substructures. So that's what lectins actually read. So for example, the lectin SNA will read 2,6-sialic acid on a complex carbohydrate. And so when we're thinking about structures that we're reading from the lectin microarray, it's really these substructural elements that you can then see detected, for example, by hemagglutinin. So they're very biologically relevant because it's not necessarily the entire structure that matters, it's really the bindable unit in biology. Next slide, please. So our current lectin microarray technology, we no longer really do single color, we do what's called a dual color array. We have used animal tissues, human tissues, cell culture, serum. We take the glycoproteins, we label them, we do two colors, so one is the sample, one is a reference that we use for that biological set of assays, so we call that a biological reference. We mix the two together, put it on the microarray, and actually read out from that a really robust semi-quantitative analysis of the changes in glycosylation across the sample set. And that makes it very integratable with other omics. We have about 100 lectins and antibodies on the array currently, and we're always expanding that using our technology. So next slide, please. So this is a snapshot. I don't want to spend time on this, but I just wanted to point out that we've used this for a lot of different stuff. So influenza is part of what we do in my lab, but we also work in cancer. We also work in women's health. We're also working a bit in COVID, so we really have a pretty broad set of things that we're now looking at the glycomic analysis of. Next slide, please. But today, I'm really going to focus on flu, and I want to point your attention to two papers that have recently come out, because the beginning part of my talk is really going to focus on the work in these two papers. So if you're interested in more detail, I'm kind of going to give a brief overview of them and hit the highlights. But one of them just recently published in the Proceedings of National Academy of Sciences, or PNAS, and the other one is in J Proteome Research. Next slide, please. So this work was done, and you can see Ted front and center in the middle, with a group of people that created the flu dynamo. So this is earlier to the civics. This is the influenza dynamic network modeling. And so it was headed by Professor Elaine, who's now at the NIAID. And of course, Bin Zhang, Ted Ross, Allison Kelvin, and David Kelvin were part of it, and Stephanie Bissell and Clayton Wiley. So this is sort of the team. Next slide, please. And we're really focusing on glycans in influenza pathogenesis. So this is kind of a slide that you may not need, but I just wanted to point out that most of what people have focused on in influenza is really sialic acid, right? It's all about the neuraminidase, which is cleaving sialicides, and of course the hemagglutinins, which are binding sialicides. So that's where a large amount of our knowledge to date has really focused. But in my lab, we really look at a more broad approach. So we really wanted to look at not necessarily the role of glycans in infection, but rather the role of glycans in determining severity. Next slide, please. So to this, we looked at the ferret model. And so what we did is we took the ferret model, and we had it, this is all done, of course, in Ted Ross's lab. We infected the ferrets at day 0 with the 2009 H1N1 pandemic strain. And then we looked to see at day 8 how they fared. So we classified mild infections by weight loss, and then moderate, and then severe. And we looked at the responses in the lung to see if we could see anything happening with the sugars that correlated with severity. Next slide, please. So this is a lot of data, and I'm actually skipping over a lot of information. But what I will tell you is that the sialic acids do not correlate with severity. We looked at that. I'm not going to show those graphs, but we did not see any association between the level of sialicides in the lung and severity from flu in the ferret model. However, if you look at the bottom of the slide, you'll notice that green box around high mannose. And if you look over at the heat map from our lectin microarrays, what you can see is an image that goes from blue to yellow as you go from uninfected to severe. What this indicates is an increase in that particular epitope, and that epitope is high mannose. And you can see the box plots on the right-hand side of your screen that show the same thing. So these are two different high mannose probes, HHL and Griffithson, both of which show a sort of increase, a very sort of almost linear increase, going from mild to moderate to severe influenza in this ferret model. Next slide, please. So we showed that the histology actually correlates with damage from the flu. So if you look at the lungs and you look at uninfected versus infected, and you look at the high mannose staining, so that's shown in red here at day 8, you can see very clearly that there's a huge amount of high mannose in the damaged lung. Next slide, please. And this actually directly correlates with the alveolar severity score. So the lower levels of HHL binding correlate with a lower level of severity, the higher levels correlate with higher. So this is nice because these are two different metrics of damage. In the earlier one, we used weight loss. Here, we're actually using a histological score, and we can see very clearly that high mannose is going with damage from the flu. Next slide, please. So, okay, so what about, let's change it up a bit. We know that age can have a correlation with severity in flu. And if we look at both young and old ferrets, what do we see? So in newly weaned ferrets, in that model system, the 2009 H1N1 pandemic 09 virus does not actually give very many symptoms. In fact, you see very mild symptoms with very little death. But still, you do get a range, and so we use the pneumonia composite score to look at mild, moderate, and severe, and to see whether we really see anything significant in newly weaned ferrets. Next slide, please. And what I will tell you is that not only do we not see high mannose significantly changed in the newly weaned ferrets, we actually don't see much of anything significantly change. And I think that's because, again, we don't really, even though we can classify it by severity, the severe in the newly weaned is not actually very severe. These animals actually don't have very many real symptoms the way that you see in adult ferrets. Next slide, please. So what happens in the elderly? So in the ferret model, so in humans, of course, the H1N1 pandemic 09 did not actually cause as much severity in the aged, but the reason was because we had pre-existing immunity. Ferrets, of course, don't have that pre-existing immunity. So the aged ferrets, when inoculated with this virus, actually end up getting very, very severe infections. And so you have very high levels of weight loss and you get a lot of death. Next slide, please. So looking in this model, and really here we had to use the pneumonia composite score because it turns out that weight loss actually happens even with lower levels of severity. So the pneumonia composite score was a more accurate measure. What we see is, again, high mannose very clearly going with severity. So the more severe cases in the aged ferrets, again, saw this really clear high mannose signature. And if you look on the left-hand side and you look at the heat map, those are actually ordered by the composite score, and you can see that you do indeed get more and more severity as you go up in the pneumonia composite score. Next slide, please. Okay, so that's in a ferret model, but what about something human? Now, clearly we cannot do these experiments in human. We can't take human lung biopsies. So we went to the cell culture model. So we looked at primary bronchial epithelia with the H1N1 virus and we infected. And what we can see is in infected cells, we see high levels of high mannose at the cell surface. And so you can see very clearly this sort of nice surface staining. And we also see this, of course, in the A549 model system. Next slide, please. And we found that it's not strain dependent. So we could use PR8, we could use ZH3N2, we could use flu B, and in all cases, high mannose came up with flu. Of course, the simple explanation for that would be that, well, it must be the influenza proteins itself. Next slide, please. So we looked at exactly that using a lectin blot. We looked at HA and NA and to see whether or not the high mannose could have a trivial explanation. And what we see is, although there is clear evidence that the neuraminidase has high mannose on it, HA it's a little bit more questionable. We also see a high upregulation of other host proteins that are all getting this high mannose signature. So we don't believe that it actually is on just the viral proteins. And in fact, in the ferret lung, it would be doubtful that it would be on the viral proteins because at day 8, you have very low viral titers in the lung. Next slide, please. So why does this matter, right? Okay, so it's a sugar, who cares? Why is high mannose on the surface? Well, high mannose is not commonly seen on the surface of cells. Normally we think about it as an epitope that's seen early in processing. We don't think of it as a cell surface epitope. And so what could the role of it be? We think it may be a signal because high mannose actually plays an incredibly important role in immunity. Many of our innate immune lectins actually recognize native high mannose, and those include DC-SINE, the macrophage mannose receptor, langerin, SPD, and the mannan binding lectin or mannose binding lectin. Next slide, please. So we focused in a little bit on the mannan or mannose binding lectin because it turns out that there's increased MBL and MASP2, so it's part of the lectin complement pathway in schizophrenics and multiple sclerosis patients. And if you look at the epidemiology of those patients, they die at a far higher death rate from influenza and pneumonia. So schizophrenics die at about a 7-fold higher rate, and multiple sclerosis patients, it's about 3-fold higher. And because of an earlier experiment from a Japanese group where they did the MBL knockout mouse and saw that they actually got less severity, a group in England actually looked at MBL levels in critical patients with H1N1. And what they found is the levels correlate strongly with mortality. So the more MBL you have, the more likely you are to die of the flu. Next slide, please. So we wanted to see if MBL would actually recognize the infected human cell. So would it look like the lectins, you know, the plant lectins that we're using when it comes to recognition? And indeed, we can show that infected cells actually show high levels of MBL binding when we treat with endo-H, which is an endoglycosidase, which removes all high mannose glycans. What we see is we see the loss of that binding, indicating that yes, MBL does recognize. Next slide, please. So due to all of this data, we actually think high mannose is a danger-associated molecular signal. that is important. So we think that this may be activating the complement cascade, and that this may be part of what's going on in severity in the flu, is that hymannose is coming up in response to infection and then causing a cascade. And people who either have high levels of mannan-binding lectin or high levels of hymannose will end up having much, much, much more severe flu. Next slide, please. So, okay, so that's kind of host response. So I want to turn for the last little bit to talking about glycans in vaccination and immunity. So this is all work that's done with the civics and of course with the Center for Influenza Vaccine Research for high-risk populations. Next slide, please. So this is the omics team of the CIVR. So I just wanted to show all of their faces. Next slide, please. And so we're doing a multi-omic analysis of sera pre and post vaccination to try to get a handle on what is it that actually may contribute to having a good or bad response. Can we see those? Can we get biomarkers? And also, can the biomarkers themselves tell us something about the underlying mechanisms? So of course, we're doing the glycomics, and there's many other omics on the way. And eventually, we hope to get a pretty comprehensive profile. But the work that I'm going to show you is from the UGA4. So participants, we're looking, the data that we have right now is on 160 people, and they received the Fluzone quadrivalent vaccine from the 2019-2020 flu season. Next slide, please. So I just want to calibrate you a little bit on the human serum glycoproteome. You see glycosylation, of course, of the immunoglobulins. You have lectins in there, glycosidases and glycosyltransferases. And I point those out because although some of them are glycosylated and some of them are not, we actually added antibodies to these proteins to the array. So some of what you will see is not carbohydrate based, it's actually sort of lectin and glycosidase based. And then, of course, you have protease inhibitors, haptoglobins, complement proteins, and many of those are also glycosylated. So you're really seeing glycosylation on a wide variety of proteins. Next slide, please. So what we're really interested in are host factors that influence vaccine response. We've already seen some examples of how biological sex can change vaccine response that was given earlier. And of course, there was some indication that maybe obesity might be something to look at. And so these are some of the things that we're really trying to look at in the CIVR. Next slide, please. So the first thing I want to tell you is that when we looked at our data set, we actually have an obese and a non-obese population. And so we actually define normal as being less than 25 BMI, obese being over 30. And when we looked at, are there changes in the glycome in the baseline that are just associated with obesity, what we found is there are indeed strong changes. And these changes are stronger in the obese versus non-obese than any changes we see with gender, although we do see some gender-dependent changes as well. So in obesity, in people who are obese, we do see higher levels of core 1O glycans, and we also see, interestingly, higher levels of sialic acids. So both the 2,3 and the 2,6 sialicides are higher in people who are obese. Next slide, please. So because of this, we really wanted to focus in on subpopulations. This does mean our numbers get a little bit low. But before we go into that, I need to also talk about defining response. So I'm sure all of you are familiar with the HAI titer scores. But I want to say that, as you can also see in the work that's been shown to date, responses are strain specific. And so when we think about that, we realize that we have to look in a strain specific manner in terms of defining people who are having a high response or a low response to a particular vaccine. And so for each strain, we looked at the seronegative non-responders. They had to be seronegative at day 0. And then if they were seronegative at day 28, they were considered a non-responder. If they were seropositive at day 28, they were considered a responder. Next slide, please. And so what we see is, if we look at the influenza A H1N1 strain, we see in obese male and obese female a very similar response, where we see lower levels of proteins actually that are correlated with immunity. So people who are responding have lower levels of proteins that indicate lectin-mediated innate immunity, so mannan-binding lectin, the ficolins, the colectins, MASP2, and they also have lower levels of IgM and IgG. It's a little gender dependent in terms of exactly what we see, but it's a very similar class of molecules that are lower in responders versus non-responders. Next slide, please. In the H3N2, we see something very similar. Again, we see lower levels of lectin-mediated innate immunity and IgG in people who respond. Interestingly, in the obese female, we start to see a glycan signature as well, which is that we see 1,2-fucose being higher in people who respond, which is interesting because there's an association between 1,2-fucose and microbiome in ways that have been thought to actually aid vaccination. So people have shown that things like bacteroides and certain strains of lactobacilli can actually help with vaccination. And interestingly, those actual organisms often use 1,2-fucose as a food source. So there may be something there, although again, these numbers are very low in terms of really getting a good association. Next slide, please. In contrast, though, if we look at the normal BMI, responders versus non-responders, and now our numbers are a little bit low because there are lower numbers of people with normal BMI in this data set, but we don't actually see anything that's statistically significant. There's no strong signature for people with a normal BMI. So we're seeing a difference in the way that people who are obese are responding versus people who have a normal BMI. Next slide, please. And the baseline, so if we look at influenza B, again, we don't really see anything clear. We see some responses that may mean something, but there's no really clear signature in the obese responders versus non-responders for the influenza B strains. And this is very different from what we see in the influenza A strains. And this is true for both influenza B strains. I'm only showing you one for time. Next slide, please. So the overall conclusions that we have from this is that what we can see is that there seems to be lower baseline lectin-mediated innate immunity in obese responders. And people have shown that adaptive immunity it seems to be better you have better response in adaptive immunity if you have lower initial levels of IgG and IgM. And it seems that the same may be true for the innate immune system. But the differences right now are only observed in the sera of obese responders versus non-responders and only to the influenza strains, arguing that we really need to take strains into account and think much more broadly about this. Next slide, please. So with that, I have to acknowledge all of my students and the civic and all of you for your attention, and I am happy to take questions.
Thank you very much for this very nice presentation, super interesting. And maybe I can start with a question. So these high mannose on the surface of cells, is that really influenza specific? Do you see that when you infect the cells with other viruses like VSV? And does this show up if you treat cells with interferon?
So, okay, so we did do early work which is not in the publication, where we treated cells with interferon, and it doesn't change that, actually. So we don't see high mannose in response to interferon, at least not in our preliminary analysis. We have not tried VSV, so I do not know if it happens with VSV. We have actually, in unpublished work, looked at SARS-CoV-2, and interestingly, it does not look the same. Of course, that may partly be the cell types, because we know that SARS-CoV-2 infection has some very interesting cell type specificities in terms of getting really good infection. So that may be a part of what's going on there, but it definitely looks like it is different. So while we don't know if it's influenza specific, and I do think that there are probably other viruses that will trigger it, it is clear that it happens in flu, and it doesn't seem to be through the interferon pathways.
And do you have any hypothesis what kind of pathway could cause it?
Yeah, so we think it's actually downstream of the unfolded protein response pathway. So the paper in PNAS actually talks about that. We did some experiments that sort of show that. And so part of that is because we had done some work with Matt Shoulders with XBP1. He has this cool, he's at MIT, has this cool system where he can turn it on and off using a chemical inducer. And in that work, we saw in a cell type specific manner, high mannose coming up in some cases with the UPR. So I think in the lung cells in this case, we looked at the UPR as a possible mechanism, and we do think that it's downstream of that. We're not, obviously this remains to have a lot of testing done, but we think that that may be one that we don't know exactly what the exact biosynthetic pathway and how it's controlled is, but we think that there's some part of that control mechanism that goes from the unfolded protein response to the high mannose response.
All right, thank you. We have a question from the audience as well. The question is, what mechanism mechanisms drive the higher core glycan expression in diabetics, and are there any comorbidities in this cohort?
So the higher core, say that one more time. I'm trying to make sure I understand the question.
So what mechanisms drive the higher core glycan expression in diabetics? And
okay.
If there's any comorbidities in this cohort?
So, you know, I am going to bet that there probably is, but I have to admit I have not looked at diabetes in flu. The core, I'm assuming they're talking about core fucose because there is a core fucose association, and I don't think that that's really known. I mean, the association between diabetes and the glycan pathways, from our own studies, I would say it's more likely to be through the transcriptional regulation systems than it is through a metabolic process itself, because we found that you can actually up levels of glucose in media to insane amounts, and until it actually starts stressing the cell, it doesn't change the overall glycome. So I think that it probably is downstream of many of the same mechanisms that cause all of the other diabetic damages, but I don't know exactly what they are in that case, and I'm not exactly sure what the association with flu is. I have to admit I'm ignorant on that one. So.
Right. Thank you very much. I think in the interest of time, we have to move on to our next speaker. But thank you very much for this very nice presentation.
Thank you.
Okay, so our next speaker is Dr. Elaine Reed. She is a professor of pathology and laboratory medicine and endowed chair of diagnostic medicine at UCLA. And she's also the director of the Immunogenetics Center at UCLA. And she's going to talk about a systems-based approach to profile natural infection and vaccine responses.
Thank you, Florian, for the introduction, and thank you to Drs. Ross and Liu for this opportunity to talk with you today. I'm going to present some of our work on using system-based approaches to profile both natural infection and some of our newer work in collaboration with the civic team at the University of Georgia and St. Jude on looking at immune correlates of influenza vaccination. So I'd like to start off first talking about some of our recent work on looking at immune correlates of antibiotic persistent MRSA bacteremia. And I think to this group, looking at vaccinations to pathogens, you probably all know that the immune response to MRSA and the ability to develop useful and robust vaccines have met with little success. So our work in this area was really to focus on trying to understand the host pathogen interactions along with the antibiotics to determine whether we could improve treatments and with an ultimate goal certainly of vaccination. The first area that I'm going to speak about, which is MRSA bacteremia, has been a collaboration between Vance Fowler at Duke University, Michael Yeaman at Harbor UCLA, and my team of investigators at UCLA Westwood. The second collaboration that I'm going to be speaking about is between UCLA and then the larger civic group headed by Stacy Schultz-Cherry and Ted Ross. Can I have the next slide, please? So most of you most likely understand the importance of MRSA bacteremia. Methicillin-resistant Staphylococcus aureus is a huge global health problem. It's striking that up to 30% of MRSA bacteremia failed treatment. And although interestingly, the MRSA isolates appear to be susceptible to antibiotics in vitro, unfortunately, they failed treatment when given to patients in vivo. And this resulting in antibiotic resistance. And the mechanisms for antibiotic persistent MRSA bacteremia are poorly understood, and clearly development of effective vaccines for protective immunity have proved to be elusive. Our central hypothesis for this study has been that persistent MRSA bacteremia reflects a unique three-way interaction between the pathogen, the host response, and the antibiotic. And we are looking at 3 different areas within this project of the adaptive response to MRSA to specific antibiotics called the persistome, the adaptive response to MRSA to host immune systems, the immunome, and the adaptive immune response of host immunome to antibiotic mechanisms. And we postulate that the host pathogen signatures of persistence can be defined, modeled, and enable predictive capability for clinical utility. Can I have the next slide, please? So our group has focused over the past few years on determining signatures of persistent MRSA bacteremia, again with the hopes that we could improve treatment. And on the right side of the slide, our group is focused on studying the host immunome, including host immune transcriptomics and host immune cell epigenetics, as well as phenotyping of host immune proteomes. So I will talk about some of this work, starting out with the host immune cell epigenetics with an eye towards trying to understand how the infection modifies the host immune methylation machinery and phenotype of the cells. Can I have the next slide, please? So there's data emerging in the literature that indeed pathogens interacting with the host immune system can modify the epigenetic machinery of the cell, causing modification of immune responses during infection. And on the far left of this slide, you can see there's influenza virus, and it's known to cause epigenetic changes in innate cells by interaction with toll-like receptors such as TLR7, which triggers downstream intracellular signaling, causing modifications of methyltransferases, which can then induce the generation of pro-inflammatory cytokines and modifications of adaptive immunity. So this modification by different pathogens can be detected by studying the phenotype and epigenetic changes in host immune cells through various different technologies. Can I have the next slide? So we recently substantiated this hypothesis that epigenetic changes are important in host immunity to Staphylococcus aureus in a study in which we looked at macrophage conferred immune memory after exposure to Staphylococcus A. And we found indeed that there are changes in macrophages following priming with Staphylococcus aureus. And this, again, substantiates that this change in immune phenotype is most likely because of epigenetic changes. Can I have the next slide, please? We further demonstrated that patients who carry a mutation in the DNMT3A gene exhibit higher methylation levels in regulatory CpG island regions. For example, patients who are heterozygous at the DNMT3A gene have increased methylation, show lower IL-10 responses, and in contrast, those individuals with wild-type DNMT3A genotype demonstrate decreased methylation responses and consequently higher IL-10 levels. And this is associated with persistent infection. Can I have the next slide, please? So our research objectives for the study were to define the differentially methylated sites, or the acronym we're going to be using in the slides is DMS, associated with persistent infection, to understand gene regulation and to understand and assess the biological functions of these differentially methylated sites by looking at the genomic landscape, the transcription factor binding motifs, and the cell type specific DNA methylation Very importantly, one of the objectives of this study was to identify classification models, to develop them, and to see if we can identify predictors of outcome using methylation, quantification of methylation. Can I have the next slide, please? So the samples, this is the study design of the work that I'm going to talk to you about, and these were samples which came from the Savage Biorepository at Duke University in collaboration with Vance Fowler, and these were human cohorts. We had two stages, one that patients were selected in 2016, and then a second cohort selected in 2017. We started out with over 3,000 cases of patients with SAB, and we narrowed that down to a very select cohort who had received vancomycin treatment and had blood cultures and biospecimens available. We found 374 in stage 1 and 496 cases in stage 2. And when we looked at complete biospecimen available samples, there were 102 cases in stage 1 and 155. We then used propensity score matching with specific covariates to control for both, because this, of course, is epigenetic studies, race, age. We also were concerned about other factors that might influence methylation patterns, and so we also controlled for patients who were on dialysis, diabetics, and if they also the device type that a patient might have had, which might have triggered the infection. So ultimately, we studied 34 pairs, 34 individuals who had persistent bacteremia, and that was described as persistent positive blood cultures over 5 days following vancomycin treatment, and the case-controlled matches for which, after the initial diagnosis by blood culture, they responded to vancomycin treatment and their blood cultures became negative. And we had, again, 34 pairs of these controls in cohort 1, and cohort 2, there were 43 pairs. Can I have the next slide, please? So again, the study design was we had chosen patients with bacteremia, vancomycin treatment, and they had two different outcomes: persistent infection versus resolving infection. We had whole blood samples in which we did RRBS methylation studies. We identified nearly 800,000 CpG sites with an average count of 15 across all the samples. And again, our goal was to study methylation across the genome. And so we were looking for gene locations, both sites and regions which were methylated. And of course, that would reflect if they were methylated, we would predict that they did not have transcription factor occupancy, where hypomethylation or demethylation would allow for transcription factor binding and then gene expression. Can I have the next slide, please? So this is the cohort and their patient characteristics. Again, because we did propensity score matching, for certain characteristics, the patients were well matched for their age, their sex, diabetes, and the type of device. So we didn't find any differences in the cohorts there. But if we then look at persistent infection, you can see that there were less cases of neoplasm in the persistent infection compared to the resolving. There were more cases of surgery in the resolving. They were surviving, or these patients with infection were post-surgery 30 days, and there were more in the resolving group. Could I have the next slide, please? In the persistent group, there were higher incidence of metastatic infection. Length of stay was greater in the patients with persistent infection. Patients with persistent infection had more surgical removal of foreign devices, surgical debridement, and abscesses. And ultimately, there was the outcome. There was increased recurrent infection and death due to infection in the persistent patients with persistent SAB bacteremia versus those with resolving infection. Could I have the next slide, please? Now, what about the methylation studies? So as I mentioned, we had identified when we looked globally using RRBS that we were interested in defining methylated sites that distinguished APMB, the persistent infection, from the resolving. And looking at a classification model, we looked at receiver-operated curves for methylation across persistent versus resolving infection. And what we found was there was a significant difference between these group of patients when we looked at their overall global methylation patterns. With the ROC showed a 76% sensitivity and 84% specificity in distinguishing persistent infection from resolving if we just looked at global differences in their in methylated DNA. We then went on to identify the, using MEthylKit, the differentially methylated sites, and we found there were 116 differentially methylated sites that were hypermethylated in AR, the resolvers versus the persistent infection, and 160 sites that were hypomethylated. On the right side, you can see the hierarchical clustering of these 276 differentially methylated sites. And we can see that these sites differentiated very effectively persistent infection from a resolving infection. In fact, 97% of the persistent patient infection on the left side, shown in the pink bar at the top, 97% of the patients were clustered according to these DMS. And on the right side, the resolving infection, 83% of the resolvers were clustered with these 276 DMS that we identified. We also found two major clusters of methylation. Again, on the upper left-hand pattern, there were 131 DMS that were hypermethylated, distinguishing persistent infection from resolving, and 145 DMS that were hypomethylated. Now, can I have the next slide, please? Oh, I just wanted to say these were independent of their race and sex, as shown in some of the bars underneath the disease phenotype. So this was really an epigenetic difference, and it was not related, we believe was related to the disease, not to their sex or race. May I have the next slide, please? So we wanted to further look at these DMS to determine whether they could improve the classification to distinguish the resolvers from the persistent infection. As I mentioned to you, this was a major goal of this project, was to determine whether we could, at the time of diagnosis of the infection, could we identify patients who were going to go on to have persistent infection. And we were identified 16 DMS that were able to discriminate persistent infection to resolving infection with a classification performance of a sensitivity of, with a classification performance of 98 and a sensitivity of 91 and a specificity of 98. Area under the curve is 0.98, as I mentioned. And if we look at the right, we see that 9 of these DMS are positively associated with persistent infection, and 7 of them are negatively associated with persistent infection. So using DMS, we could really develop a model that we could distinguish persistent infection and resolving infection right from the time that the patient was diagnosed. Next slide, please. Now, what are the characteristics of these DMS? Do they bind to transcription factor sites? and what genes are they putatively regulating. And so we wanted to determine the putative transcription factor binding sites for these hypomethylated sites in both persistent and resolving infection. And so we used locus overlap analysis to overlay the DMS with previously identified validated chip sequence peaks. And we were able to identify a number of different binding sites for transcription factors which are known to regulate the immune response, in particular the immune response during infections. We identified binding sites for CEP beta, which were decreased. So hypomethylation was increased in persistent infection for these imputed transcription factor binding for CEP beta. We also found binding sites for STAT1, again, another important transcription factor regulating Th1 responses. In the next slide, please, we found hypomethylated sites in resolving infection, and these were binding sites for glucocorticoid receptor, as well as P300, which both of them are known to regulate granulopoiesis and together histone acetyltransferases. So we were pretty excited when we found these and were able to identify these sites, because we've identified transcription factors which truly have been previously identified in other infections as well as MRSA bacteremia for regulating the immune response. Could I have the next slide, please? So we went on to confirm this work using a targeted bisulfite sequencing method. We just developed this over the past few months in the laboratory to confirm these disease-associated methylation signatures. And so these were targeted probes that spanned these methylated regions and sites that we identified. And we developed over 1,000 different probes. And interestingly, because these were sequenced, we were also able to examine genetic polymorphisms with these regions and rule out that any of these associations we found between the methylation signatures and disease association were not due to genetic polymorphisms. And what I'm excited to say is that we were able to confirm, using the targeted bisulfite sequencing, the binding sites for CEP beta, for STAT1, and for GR and P300. And if we use the TB, these 121 DMS that we were able to confirm by targeted bisulfite sequencing, and again, model a classifier, we had an area under the curve of 0.9. And this is very comparable to the RRBS technology, in which, again, we had an area under the curve for a classifier of 0.97. And up the upper right-hand panel, you can see there's a excellent correlation between the results obtained between these two methods. Can I have the next slide, please? So I'd like to summarize this part one, and we described for the first time that DNA methylation profiles of circulating immune cells in whole blood collected at the time of diagnosis of Staphylococcus A bacteremia are predictive of effective or ineffective immune control of SAB. Hypomethylation of certain transcription factors, including CEP beta and STAT1, are associated with persistent infection, and hypomethylation at the glucocorticoid receptor and histone acetyltransferase B300 binding sites are also associated with disease phenotype. These are key transcription factors in emergency granulopoiesis, for example, for CEP beta, STAT1 binding in hyper TH1 polarization, and we predict or likely suppressing a protective TH17 polarization response. And again, for the GR, most likely important in granulopoiesis. So this work provides a proof of principle that DNA methylation signatures within the host immune cells can predict APMB. So I'd like to go to the last part and briefly talk about our work. Can I skip 2 slides? I might be close to running over. Can I continue? Go forward, please. I'm going to just go to the last part. Can I go to the next slide, please? Next slide, please. Next slide, please. Okay, next slide. This is the group that worked on the MRSA project, and the lead is Michael Yeaman in the middle, and Vance Fowler is all the way on the right in the back, on the left in the back. Can I go to the next slide, please? So I just briefly want to mention this work that we're doing now with Ted Ross. For the Center for Influenza Vaccine Research for High-Risk Populations. Our role in this project is looking at the immune response to vaccination, both to existing seasonal flu influenza vaccinations, as well as to the new vaccines that are being developed by the team. Our key questions are: what are the factors for vaccine effectiveness in different populations, and whether pre-existing immunity to influenza affects vaccine response. Can I have the next slide, please? And we've been specifically focused on looking at immune correlates of vaccine response to Fluzone in the UGA4 cohort. This is a cohort of patients that received seasonal vaccination. This is the quadrivalent Fluzone that was given to these volunteers, healthy volunteers, in the 2019 seasonal flu, and we were able to profile these patients or these healthy controls to look at their cytokine response over the course of the vaccination pre and over the 28-day follow-up following vaccination. We've also doing other types of systems immunology approaches, but I'm only going to report to you today on some of our initial findings of looking at cytokine responses. So we use a 38-plex Luminex assay to quantitate circulating levels of cytokines and chemokines in the circulation of the vaccinated individuals. And again, this is a longitudinal study. And we're using as immune correlates of immune response, seroprotection and seroconversion. Seroprotection, the classification is to have a certain HAI titer, which is defined by Ted's team as having a titer of greater than 1 to 40, and seroconversion being a 4-fold change in titer, HAI titer from baseline to day 28. So interestingly, we've been able to identify in this very early part of our study some cytokine differences, both at baseline and at day 7 following vaccination, that correlate with a seroprotection response. I do want to say at first, age and sex are associated with both baseline as well as day 7 cytokine responses. So we use those as a cofactor and corrected for those in all of the analyses I'm going to speak about. But maybe not surprising, females had more higher levels of particular cytokines and chemokines than males, and age was also a factor. Older individuals, higher levels of cytokines and chemokines. So this table just shows a change in cytokine chemokine levels at pre-vaccination. Again, we just looked at baseline at the time when they got their vaccination, and we find that MCP1 or CCL2 is predictive, as well as MIP1 beta is predictive of seroconversion. and seroprotection. Can I have the next slide, please? I think the box and whisker plots are presented here. As I mentioned again, this is a 38 chemokine cytokine multi-parameter analysis. And again, we've identified CCL22 as having increased levels. And the increased levels were also correlated with the number of strains they were seroprotected against, as well as GCSF. The lower the GCSF level at the baseline, the higher number of protection against the number of different strains within the vaccine. May I have the next slide, please? We found pre-vaccination levels of CCL22, MIP1 beta, and eotaxin correlate with post-vaccination seroconversion. Any seroconversion event or the number of seroconversion, again, higher levels of CCL22 and CCL4 correlate with seroconversion, and lower levels of eotaxin, eosinophil recruitment is associated with the lack of seroconversion to the number of strains. Can I have the next slide, please? What about day 7 post-vaccination differences? Again, we found 2 interesting, maybe not unexpected findings. IL-17A were higher in individuals who were seroprotected at, say, day 28 at day 7, as well as interferon gamma and IL-17A in individuals who seroconverted. Next slide, please. This is the box and whisker plots showing the levels of IL-17A, interferon gamma, and which are related to both seroconversion events and seroprotection events. Next slide, please. We put these univariate into a multivariate analysis and found that both MDC and interferon gamma are still both predictive of seroconversion and seroprotective events at day 28. All models, again, included a covariate's age and sex. Next slide, please. So in summary, age and sex impact the human antibody responses to influenza. Pre-vaccination levels of CL22 and GCSF correlate with post-vaccination seroprotection. These cytokines, MIP1 beta and CCL22 and eotaxin, correlate with post-vaccination seroconversion, and increases in IL-17A and interferon gamma at day 7 are associated with seroconversion against one or more flu vaccine strains. While immunization stimulates the production of protective antibodies, we need far more studies to determine if immunization promotes the generation of Th1 and Th2 specific T cells, and how cellular immune responses are contributing to the protective immunity. So my last slide is just the acknowledgement of the teams that participate in this study. Thank you for your attention.
Thank you very much. Unfortunately, you're way over in terms of time, and I'm going to hand it back to that to close the meeting.
Thank you very much, Florian. Thank you, Elaine, for the presentation. It was really nice. I want to first thank my colleagues, Tony Moody and Florian Krammer, who actually run 2 of the civic vaccine centers, and appreciate their time today to host sessions along with me on this symposia on updating for the influenza civic program for North America. I'm hoping that we'll be able to do something like this again in a few months to see some updates. I want to let you know that the civic program involves 7 different organizations, sites, the 3 you heard about today, but also Kathy Newsel heads up the clinical core at Maryland, and there's also a clinical core at Duke, along with a manufacturing core associated with the civic. And so you've really just seen a tip of the iceberg of the number of investigators associated with this. Each one of the individual sites has between 10 and 30 principal investigators that work on this study together. So we've really just shown you a snapshot today of the potential number of individuals that are working on this project. And so in the future, we might be able to do this again and highlight additional presenters. But I want to say thank you all for attending today. and we hope to see you again in a virtual Congress in the near future.